Hello. Okay. Nope. All right. Let's screen one. Okay, cool. All right, now you should see me in the corner. All right, chapter 8. Okay. Very difficult chapter. Okay. I'm gonna try to simplify this for you guys a little bit, but again, it's one of the more complicated chapters. These middle chapters are actually by far the most complicated ones of the lecture of the course. So we can go through it slowly and stuff, but let's just get through it. Okay. Okay. So the first part, the organization of the nervous system, you should really kind of already know. So I'm gonna skip this ahead. Make sure you're aware of what all these things actually mean, because I'm going to use these terms, okay, a lot in the next couple of chapters. Okay, so organization, you should already know that. Okay, neuron structure, you should already know that. Okay. These you should already know. So again, I'm just flipping through these slides. If you don't know it, go back to your anatomy stuff. We explained it there. If you don't have like Zoom lecture recordings of the previous anatomy instructor, let me know. I'll give you mine. But you should know what all these things are already. Okay. But you should know what all these parts are already. Okay. All right. Okay. So axonal transport. Okay. First of all, neurons are very metabolically expensive. Okay. We spend a lot of energy maintaining neurons. Okay. Your brain is about 3 pounds. Your brain consumes about 20% of all the energy you use every day. Okay. That's why humans have to eat more than most other species, because we consume so much energy with our neurons. And the reason why we consume so much energy with our neurons is this. Okay, so you have a neuron cell body, you have axons and dendrites. The neuron cell body makes everything, but you have to ship everything up and down the axon. And then you have all these sodium channels that are pumping things in and out of the axon. Okay, so it's really metabolically expensive. Okay, so axonal transport, you can move organelles. You need mitochondria at the very ends of the thing, so you have to pump mitochondria all the way down and then back, etc. So it's expensive. Okay. All right. So here's the task. Axonal transport. They're actually tracked along these freeways that travel down the axon and then travel back. Okay. Okay. So they're used, they travel back, and they're destroyed, and they're remade, and travel forward and travel back. So really metabolically expensive. Okay. Okay. So establishing synapses. Okay. So synapses change over time. Okay. How big a synapse is, like where a synapse is, what a synapse is with, etc., etc. Can change over time, especially when neurons are damaged and stuff like that. So, yeah, there's a lot of neuron development going on. Okay. All right. So, there's another video. Actually, I must play this one. Screen share sound.
As you have seen, concentration gradients drive sodium ions into the cell and potassium ions out of the cell. However, potassium diffuses.
Actually, I'm gonna come back and play this later when we talk about this stuff later. Okay. So glial cells and their functions, you should already know from anatomy. So if you don't remember what it is, please review that. Okay. So this is what this is actually a more realistic picture of what the glial cells actually look like and how they're related. Okay. Okay. So please understand how myelin works. We explain that to you guys in anatomy as well. If you guys don't remember, go back. And if you don't remember that, then ask me in class. Okay. Because we need to move forward and stuff like that. Okay. So stem cells. Okay. Okay, so rule number one for neurons. Okay, neurons can regenerate axons, but it cannot regenerate a cell body. So the cell body dies, the neuron dies, and that's it. Okay, if it's a motor neuron, okay, then what happens is the target muscle results in permanent paralysis. Okay, and you see this a lot, like in one groups of people who are actually suffering from this now is old people who used to get polio. Okay, so the polio damages neurons, but in young healthy adults, usually it doesn't damage that many neurons, and without loss of that many neurons, it's not that debilitating, and so they're functionally fine until they hit old age. Now you hit old age, and you have more neuron degeneration, and all of a sudden it matters a lot now because you have very few neurons, and so or you have fewer neurons, not very few, but now a greater percentage of neurons are damaged, and you can get partial or complete paralysis because you had polio many years ago. Okay. Okay. If only the axon is severed, then the cell body can and the severed portion can regenerate. It takes a very long time, but it does come back. If it's if the cell body dies, then it disappears. Okay, sensory neuron, you lose sensation from an innervated area. Okay, like I lost sensation half of my lower jaw for a long time because it damaged the sensory neuron surgery, wisdom teeth extraction, and therefore like I couldn't feel anything for a while, but it did grow back. Okay. So here's the repair. So like if you damage the axon, okay, you can repair it. So the proximal axon will swell. It will like macrophages and neutrophils will destroy all this stuff downstream, and then that will grow into a new axon terminal, and then it will just grow out and eventually get back to where it was. Okay. So now, okay, axon signals and neurons. Okay, again, you learn the nurse equation. The nurse equation really calculates the potential for that one ion. The sum of all the nurse equations will give you the membrane potential of the neuron. Okay, but I'm not holding you guys to using this as a calculation. Okay, but you should know what the nurse equation actually tells you. Okay, so there's two things that that tell you what the nurse equation, what membrane potential is, and that's concentration. So concentration on inside and outside. Okay, and how much is it? Okay, and then the membrane permeability. The membrane permeability will affect the ratio. Okay, and last thing, the Z is charge. Okay, so some things have a +1 charge, some things have a -2 charge. So you have to put in those values and see. And I'll tell you the membrane potential. Okay. So you can see that this and the Nernst equation, this is just a sum of the Nernst equation. Okay. All right. So this video, I want to make sure you guys see.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, hopefully that makes sense. So remember, the most important thing is the resting membrane potential is -70. We're going to play around with that now. So, 70 is the default, okay? Okay, so there you go. Okay. You guys can calculate the membrane potential of calcium, and you realize there's a lot more calcium on the outside than on the inside. Okay. And you'll see which way the charge goes. Okay. 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 So again, this is the default membrane potential, -70, the resting membrane potential. Okay. When it goes up, okay, when it moves toward 0, it is depolarizing. Okay, so polar is a difference. So depolar means removal of that difference. So depolarizing. When I increase the difference, I'm hyperpolarizing. I know it's going more negative, and therefore you think it's depolarizing because it's going more negative. But that's hyperpolarizing because you're making it more the difference greater from 0. Okay. 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 Ion channels are named for the primary ion that passes through them. that also means other ions can pass through them, just not as much. Okay, like sodium can pass through potassium, or potassium can pass through sodium channels a little bit, not and stuff like that. So, okay. All right. The voltage obeys something called Ohm's law. Okay. Ohm's law is the difference in volts is the ion flow times the resistance. Okay. So you increase ion flow or you increase resistance, you get more voltage. Okay. Okay. But if you maintain a voltage, then if one goes up, the other must go down. Okay. So what you want really is to decrease resistance and therefore increase current. Okay. 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 So there are two basic types of electrical signals. Okay. One are gradient potentials. Gradient potentials are very like the way I think about gradient potentials, you don't all get the same grade. Okay. So gradient potentials are graded, which means they're variable in strength. Okay. They're also variable in the type of signal coming through them, whether it's a positive channel signal or negative signal. Okay. They're great for short distance communication within the neuron, but actual potentials are very brief but very large depolarization. And I will explain how large that is in a little bit. But that large depolarization can cause things to happen in rapid succession downstream. So you have active potential that keeps propagating, okay, down the neuron. So we'll talk about how that works in a little bit. Okay, so make sure you know the difference between gradient potential and action potential. Okay. All right. So like everything here is going to be important. Okay. The only thing you really not need to know are ions involved, but I don't care about that one, but all the other ones are important. So make sure you go through all that. Okay. All right. So let's take a look at how gradient potential works. Okay. Okay. So here, okay. So we'll look at the cell body of the neuron. So here's a neuron. There's going to be a stimulus. Okay. That stimulus can either activate activate a sodium channel, or activate a potassium channel, or activate a chloride channel. Okay, in this case, it activates a sodium channel. A sodium channel, remember, sodium is on the outside, potassium is on the inside. So when you activate a sodium channel, it allows sodium to rush in. So if positive charges rush into the cell, this area is a little bit more depolarized. Okay. Okay. So I depolarize that area a little bit. Okay. So again, I can also use potassium channels. Screwing those two up. potassium channels, remember potassium is on the inside. If I let the potassium channel open, then the signal rushes out and I hyperpolarize the cell. Okay, so what I'm trying to get to is this area right here. Okay, so let me explain one last concept here before I end this. Okay, so this here is the axon hillock. Okay, the axon hillock will determine whether or not that axon fires or not. Okay, I have these potentials, graded potentials happen all over the cell body. I add them all up at the axon hillock. Okay, the graded potentials can be either excitatory or inhibitory. Okay, I add those all up at the axon hillock. Okay, so I have something called a summation. It all adds up. All the pluses, all the minuses, all add up at the axon hillock. And if I get over if I get over the threshold is minus 55, I fire. Okay, if I don't get over the threshold, I don't fire, and nothing happens. Okay, so ultimately, okay, a neuron can only do one thing. It can either fire or not, fire or not fire. Okay, if it's sub-threshold, then it doesn't fire. If it's above the threshold, then it does fire. All right, hopefully that makes sense. I'll try to re-explain again part 2. Okay, all right. Have a good day, everybody.